Good morning, everyone. I'm Liz Fernandez with Professional People Training, and I would also like to welcome you to today's webinar. Our speaker today is Dr. Fawzi Mohamed. Dr. Mohamed is a Veterinary Medical Officer with APHIS at the Foreign Animal Disease Diagnostic Laboratory at the Plum Island Animal Disease Center. Dr. Mohamed graduated from the University of Khartoum, Sudan, with a veterinary medicine degree in 1982 and worked as a veterinary medical officer in Sudan. He completed a veterinary pathology residency program and received his MS and PhD degrees in veterinary pathology at the University of Georgia. He also completed a postdoctoral research program at the University of Georgia. Dr. Mohamed worked as a senior resident at the Department of Veterinary Pathology, University of Florida, and lecturer at the Department of Pathobiology, Texas A&M University College of Veterinary Medicine. And with that, I'm going to turn the webinar over to Dr. Mohamed.
Thank you, Liz, and thanks everyone for joining us today. I'm glad to be here. I'm talking to you from Plum Island, New York. Today we're going to talk about Rinderpest and PPR. These are two very important diseases. Rinderpest kills more cattle than any other disease in history. And PPR is also very important. It is the most economically important disease in Africa, Middle East, and Asia. And it's a major obstacle to agriculture in these countries. So as you know, you all know by now, Rinderpest has been eradicated in 2011, which makes it the first animal disease to be eradicated, and also only the second after smallpox. So this was really a success story, the eradication of Rinderpest, and we're going to talk briefly about that. The OIE and the FAO has actually also set the goal of eradicating PPR by 2030. This has been announced in 2015, so it's been like 6 years on this program, and it looks like it has very good effect so far. So in this presentation, we're going to talk briefly about these two diseases together because they share a lot together. They're very similar viruses that cause these two diseases. And then we are going to focus in Rinderpest and then talk about PPR, and then at the end we are closing by talking about diagnostic criteria, which are also very similar. So this is the virus. It's a morbillivirus, and as you can see here in this electron micrograph, it's a negative staining, and it has an envelope. Mostly it is a round virus, but it can also be filamentous. The envelope is important because it makes disinfectants, a lot of disinfectants being effective to inactivate this virus. The virus family is Paramyxoviridae, genus morbillivirus. And recently, actually, the International Committee of Taxonomy of Viruses has named these viruses Rinderpest morbillivirus and small ruminant morbillivirus. There is 3 lineages for Rinderpest morbillivirus and 4 lineages for small ruminant morbillivirus, which is PPR. But the good news is that these are all considered just one serotype. As you could imagine, that makes vaccination very easy to manage. It's not like foot and mouth disease where you have 7 different serotypes and more than 60 subtypes, which is make it harder to design a vaccine. But here's only one vaccine can work for all lineages for each disease. Also, there is some variability in the virulence, so some isolates will be more virulent than others. So what other morbilliviruses we know of? They are all very famous diseases: canine distemper, measles, and marine morbilliviruses. And all these viruses are really very significant in their respective species. when they happen, they cause really high mortality. And recently, actually in 2012, feline morbillivirus has been also described, but it's mainly causing, been associated with chronic kidney disease in cats. So this is how these morbilliviruses are related to each other. This is showing the phylogenetic tree. And as you can see that the PPR and Rinderpest are closely related, but as they are now different identities, these are two distinct viruses. Measles is also very close to this group, and also we have the cetacean morbilliviruses and then the carnivores morbilliviruses. Since Rinderpest is the oldest disease known, actually PPR and measles have been thought to be kind of evolved out of Rinderpest, but for now they are very distinct viruses. So transmission of these two diseases occur by direct contact with the respiratory and lacrimal secretions, and actually I could say all outbreaks have been associated with the introduction of a live infected animals. These viruses are very fragile and very sensitive to heat and sunlight, and they don't really survive long outside of the host, only for a few hours. Under favorable conditions, they might survive up to 3 to 4 days. The virus can actually also persist in meat that is frozen or chilled before rigor mortis. Because rigor mortis kind of change the pH and kills the virus. So if it is frozen meat, this virus can survive several months. If it is chilled, it can survive up to 30 days. As I mentioned earlier, several disinfectants are effective in disinfecting this virus. Kind of a list of them. Another important point that I want to make is that carrier state does not exist with these two diseases. So the virus either kills the host or it just disappears. So it doesn't really hang on into the host for a long period. This is very different than, again, foot-and-mouth disease, where in cattle the virus can persist up to 3 years. But with these two diseases, the virus doesn't really hang on. It just kills the animal or it disappears from the blood. So this kind of makes eradication easy as well. So the pathogenesis of these diseases are very similar. So the virus enters the body through the respiratory tract or oropharynx, replicates in the tonsil and regional lymph nodes, and then it is spread by viremia to lymphoid and eventually epithelial tissues. There is two receptors that have been known. The SLAM receptor, which is also called CD150, is called signaling lymphocyte activation molecule, and is present in lymphocytes, macrophages, and dendritic cells. This was found to be a receptor for these viruses, and also recently they described a Nectin-4 receptor in epithelial cells contribute to the pathogenesis of these diseases. So the virus has a core affinity to lymphoid tissues and secondary affinity to the epithelium, the gastrointestinal tract, respiratory tract, and urogenital tract. So with these diseases, you see lesions on these tissues, gastrointestinal, respiratory, and also urogenital tract. These viruses are known to cause lymphopenia and immunosuppression, and often they activate latent infections like hemoparasites, for example. In cattle, when they are immunosuppressed, these latent infections they show up. With PPR, we often see secondary respiratory bacterial diseases, aspergillosis as well, which can actually sometimes kind of confuse the diagnosis as well. So this is the host range. As you can see, Rinderpest is the primary disease of cattle and buffalo, where PPR is a disease of the small ruminants, mainly goat and sheep. Pigs are susceptible, and actually they found that the Asian pigs are more susceptible than the European pigs, which show a subclinical disease. Cattle and swine will have a subclinical disease with PPR. These two diseases also affect wildlife, and this is actually very important. The African and Asian game animals, giraffes, kudus, wildebeest, and in fact, Rinderpest kind of caused a huge dent into the wildebeest population, and when the disease was eradicated, this population of wildebeest came back. It was very obvious because of Rinderpest. With PPR, actually within the last decade, they discovered more wildlife affected by this disease. White-tailed deer, the American white-tailed deer, were found to be experimentally susceptible to PPR. And then, of course, they have the gemsbok, the gazelles, and actually some outbreaks, it caused like 100% mortalities in gazelle in the Middle East. Other affected animals include Nubian ibex, Leicester sheep, impalas, and also recently in 2017, an outbreak was described in the Mongolian Yaga antelope, where it caused very high mortality, actually high, like half the population of these animals, which are endangered species, has been killed because of this disease. It's very significant in wildlife. So now I'm going to focus on Rinderpest. As you know, and we talked about the eradication program that has been accomplished by 2011. This disease is an ancient disease. It's known since the 4th century AD in Europe, and it's thought to be originating from Asia. In the 1800s, it killed most cattle in Europe, and then it was introduced into Africa in the 1895 and caused what they call the Great African Pandemic, where 80 to 90% of cattle and wild ruminants died in sub-Saharan Africa. It caused famine in Ethiopia, resulting in the death of 1/3 of the human population, and that's because people depending on cattle for their meat and milk, but also they use cattle for farming. In 1920 and 1923, it has been introduced into Brazil by Zebu cattle from India in 1920, and in 1923 into Australia. But it has been eradicated quickly by stamping it out. This of course never occurred in the U.S. So these are very interesting historical landmarks. The concept of quarantine and stamping out methods that are made by Lancisi in 1715, and he's the personal physician of the Pope, and as you know, at that time the veterinary profession didn't even exist. So he was consulted by the Pope to control or figure a cure for this disease, and he suggested stamping out a protocol, which is mainly killing all affected animals and all animals in contact, and very thorough disinfection of infected animals. So this exists today. As you can imagine, people till that time they were using different kind of cures. Of course, it never worked, but the stamping out of protocol is kind of adopted since that time, and it's used even today. So in 1762, the world's first veterinary school opened in Lyon, France, and that's actually mainly to study Rinderpest and teach these Lancisi methods of stamping out. In 1865, the clinical thermometer was developed. This is both for human and veterinary medicine. In 1902, Rinderpest was shown to be caused by a virus, and also in 1924, the OIE was created, and mainly to combat Rinderpest. This is very, very rich history. So the clinical signs of this disease of Rinderpest can be summed as the 4 Ds: depression, diarrhea, dehydration, and death. And you can see here this is an infected cow that's having really watery diarrhea, and these animals get dehydrated very quickly and they die within 8 to 10 days. Really very fatal disease. You could also see these mucosal eruptions in the mouth, and they start with just small, sharply demarcated erosions. They call them raised pinheads because they are very small, and then they coalesce, they become bigger erosions, and they cause spotting of epithelium. Often these animals will have secondary bacterial infections, which kind of cause the bad odor or halitosis that is being described with this disease. Sloughing of the mucosa, as you can see in here in this hair palate, and also in the dental pad over there. So this will be good actually to collect for diagnostic purposes. Another area where you could see lesions is the base of the tongue. You can see erosions in there as well. And also in the commissure of the mouth, you can see these small erosions and also planting of the dental papillae. So since this virus targets the lymphoid tissue, this is the Peyer's patch, and you can see these Peyer's patches are necrotic and covered with this fibrinous pseudomembrane. And this is very characteristic lesion when you see it. Another region that you can see lesion is the ileocecal valve, and this is showing again areas of necrosis and membranes, fibrinous pseudomembranes attached to the necrotic and eroded areas. So for differential diagnosis, diseases that can be confused with Rinderpest is BVD or mucosal disease, IBR, infectious bovine tracheitis, malignant catarrhal fever, vesicular diseases, stomatitis, or foot-and-mouth disease, although you don't really see the diarrhea with these vesicular diseases. The difference with Rinderpest is that Rinderpest is really fast-moving, can cause really high mortalities and very high morbidity. BVD also is kind of affect 1 to 2 years old, where Rinderpest can affect all ages. So a little bit about the stamping out or the eradication of Rinderpest. This is one of the posters that is used by the OIE and FAO, and here stays working to stamp out cattle plague by 2010. Actually, that was very close because it was eradicated by 2011. So, of course, Rinderpest eradication is by itself is a huge story, and several international organizations have contributed to the control of Rinderpest, which eventually finally leading to the eradication. There are several organizations that worked on this over the years, sometimes because of wars or political arrests. They haven't really completed the job. But the big push came where the global Rinderpest eradication program was created in 1998, and it's backed by the FAO and OIE. And it's made with the goal of eradicating the disease by 2010. And of course, they have huge strategy that is being followed, and actually now is being related to do the same for PPR. Finally, of course, Rinderpest has been declared eradicated in May and June of 2011. But the last reported case was actually in Kenya in in 2001, just almost 20 years ago. The last 10 years actually was basically verifying and making sure it has been eradicated. The last use of Rinderpest vaccine was in 2004. So this kind of show you the stages that eradication program went through. In the 80s, as you could see, the disease is still very spread in Central Africa and also Middle East and Asia. When you come to the 90s, as you can see, the disease kind of disappears from West Africa, mainly because of vaccination campaigns, but it still existed in East Africa, Middle East, and Asia. In 2001, there is 3 spots that still infected, one in South Sudan, and one in Pakistan, and one in what they call the Somali ecosystem. This kind of mostly disappeared. In 2004, there was a spot in the Somali ecosystem that was suspicious, but actually turned out to be just an antibody response in an older cow. The disease has been really, the last case has been confirmed by 2011. And then in 2009, as you could see, the disease completely disappeared. Surveillance has continued, and every country has to submit a document verifying their freedom. And of course, that has paved the way to the Rinderpest eradication announcement by 2011. So the OIE and FAO put a global Rinderpest action plan post-eradication, and it was very detailed, and it's published in 2018. So they have designated 5 labs as Rinderpest holding facilities where Rinderpest containing materials can be sequestered. Other labs were either to destroy their holdings or submit them, transfer them to one of these holding facilities. There is 2 labs that have been designated as vaccine repositories and also as to manufacture vaccine if needed. Research with live Rinderpest virus is now banned. This can only be done in highly controlled experiments and only supervised by the OIE and FAO. So progress really has been made now in sequestering the virus, and no reports of Rinderpest have been since the eradication has been declared. Actually, the last case, as I mentioned, it was 2001. So this leads us now to talk about PPR. This is distinct disease, as you could see at the beginning. Actually, when PPR was described in 1942, there was a lot of confusion. Is this just a Rinderpest in small animals, or is it a different disease? It was found, of course, it's a different kind of disease, but very similar to Rinderpest. Other names used for this disease are goat plague, stomatitis, pneumoenteritis, which kind of sums up the clinical signs you see with this disease: stomatitis, pneumonia, and enteritis. Also called pseudo-Rinderpest or catarrh, from catarrhal rhinitis that has been described. So this disease, as I mentioned, it was first described in West Africa, in Ivory Coast, in 1942, and it stayed in West Africa actually for a long time, till like 1970s, where it pops up in Sudan, and then after that it kind of spread in other areas in Middle East and Asia. The disease now occurs in Africa, Asia, Middle East, Turkey, and it's been reported in China in 2007. And as you can see, it's kind of spreading in the last decade or so. Tanzania, Tunisia, Algiers, Congo, Angola, Mongolia in 2015, and then in Georgia in 2016, and Bulgaria in 2018. So it's kind of spreading to new regions that has not been affected in the past. As I mentioned earlier, there's 4 lineages known. 1, 2, 3, and 4. 1, 2, 3 all mainly occur in Africa, while lineage 4 occurs in Asia. Most isolates currently belong to lineage 4. So the clinical signs of this disease, PPR's incubation period, range from just 3 to 15 days, but average about 4 to 6 days. It starts with depression and fever, 104 to 106, and then you'll notice congested mucous membranes and necrotic stomatitis 1 or 2 days after the onset of fever. They will have serous and then mucopurulent nasal and ocular discharges, and then severe diarrhea about 2 to 3 days after the onset of fever. So the diarrhea will start soft and watery and then become foul-smelling and blood and necrotic epithelium. It's really severe. Later in the disease, they will have increased respiratory rate because of the interstitial viral pneumonia, coughing and sneezing due to pneumonia. And this is a big difference between PPR and Rinderpest, where with Rinderpest we didn't really see the respiratory component of it, but with PPR they will have a truly viral interstitial pneumonia, coughing and sneezing. Well, these animals will die because of dehydration within 7 to 10 days. They could also die because of the pneumonia and the secondary infections as well. So I'll show you some pictures here. This is a goat. As you can see, the mucopurulent nasal discharges and also lacrimal discharges, as you see in this goat. In the mouse, you can see these erosions of the mucosa, and this is like a cheesy white material covering the mucosal surfaces, which is mainly slough epithelium. And then also this erosion you can see also on the tongue, the surface of the tongue, and this is very characteristic for this disease, also in the dental pad. Later in the disease, you could see these papules on the commissure of the mouse, which look very similar to parapox or contagious eczema. Again, this virus, of course, targets the lymphoid tissues. This is first patch, hemorrhage, and necrosis. We see this disease. And also, you can see small erosions, which is very hard to see in this one, but you can see that is the congestion of the colon. But this can also have erosions in that area. Often described with this disease what they call zebra stripes. They're also described, of course, with Rinderpest. They call zebra stripes or markings. And mainly hemorrhages and erosion along the mucosal folds of the colon and rectum. So these are not pathognomonic for this disease. This is commonly described. And as I mentioned earlier, these animals with PPR will have a truly viral bronchointerstitial pneumonia. Often they will also get secondary bacterial infection with Pasteurella. Of course, the name has changed now to Manhamia and Leverstinia. So it's this bacterial infections that often really confuse the disease. So you find an outbreak and you kind of isolate the bacteria, and often forgetting that the primary cause is actually underlying viral diseases. So histologically, what you see in the colon, this picture here is necrosis of the crypts and inflammation. If you I was actually trying to show you the inclusion bodies. This is where I can't show that for some reason. But anyway, this is the lung section where you can see also a big syncytial cell. This is the interstitial pneumonia, and it's truly viral pneumonia. But with this disease, you see the syncytial cells, these intranuclear inclusion bodies. Inclusion bodies can also be intracytoplasmic as well, both intranuclear and cytoplasmic, and can be diagnostic. So the morbidity is very high, especially in naive population. It can be up to 100%. Mortality can range from 20 to 90%. In endemic countries, mortality can be really low, only 4 to 5%. Recovered goats and sheep become immune for reinfection. There is a PPR vaccine, of course. We're going to talk about that a little bit later. It protects animals for up to 3 years and maybe for life. Seroprevalence can reach up to 50% in some countries. And this indicates that in these endemic countries, the disease can really become clinical and not really detected unless you're doing lab testing. So differential diagnosis for PPR, this is a long list: contagious caprine chloropneumonia, pasteurellosis, which all these can be the secondary infection, contagious eczema, tongue, foot and mouth disease, parasitic enteritis, bacterial enteritis, anaerobic sheep disease, and toxicity. These are all differentials for this disease. So this kind of show you how this disease spread. It started, as I mentioned earlier, in West Africa, in Ivory Coast, where it was described, and then it spread to Sudan in 1972. Then it went east, appeared in India. By 2007, it was kind of like really widely spread in Africa, Middle East, and Asia. In 2008, it gained even more ground. And in 2012, it spread even more south Africa and north also. It's kind of spreading to different areas, and actually this could be just because people are becoming aware of it and finding it, or it could be actually truly spread, which is actually was could happen at the same time. So currently, this is more like what the current situation is. The red areas are infected areas, as you can see. It's spread all over Africa. South Africa is still free. The yellow areas are at-risk areas, and the blue areas are free areas. So this is still widespread in different countries. So, encouraged by the eradication of Rinderpest, OIE and FAO have set the goal of eradicating PPR by 2030. That was declared in an international meeting in 2015. It's been now 6 years on this program. To know that the global small ruminant population is about 2.1 million, but more than 90% of that is in Asia and Africa. So eradicating these diseases really be very helpful to these areas, and especially for poor families which rely on small ruminants. Maybe about 300 million poor families they rely on small ruminants, which and this has a huge impact on the animals. So there is a global strategy for the control of PPR, as published in this document here, and it's mainly following the strategies that are used for Rinderpest eradication. So this kind of outline, I'm not really expecting you to be able to read this, but it just shows you there is a strategy that is lined out. So there is 4 phases to it. Phase 1, mainly preparation, about 3 years, and then phase 2, which is the eradication phase, about 5 years, and mainly consists of control and vaccination. And then accreditation, which is about 4 years, that takes every country to verify their freedom. And then the post-eradication period, which is mainly to gather all virus and sequester it in one place to avoid any reemergence. And each country goes through these phases separately. So some countries might find that they already did phase 1, they just jump to phase 2. So every country will have a different program to follow and different timeline. So far, of course, as I mentioned, we are in this eradication period, and the outbreaks number has been less. Actually, as I said, the reporting of the outbreaks, especially in Africa and certain areas, is kind of less outbreaks than before. So some progress has been made. So finally, I'm going to talk about the laboratory diagnosis. So from live animals, the samples that are favorable for the diagnosis are conjunctival fluid. So you take a swab from that conjunctival fluid, as it can be good really for PCR or even virus isolation. Epithelial tissues, as you could say, this disease can cause a sloughing of the epithelium, so it's very easy to gather it with the swab. You can use whole blood for virus isolation using the Buffy code. You can also use the serum, of course, to test for antibody, lymph node aspirate, or fecal material. From dead animals, you can collect intestine, tonsils, spleen, lymph nodes, and a set of formalin-fixed tissues. So in the lab, we have different tests we can use for antigen detection or antibody detection. So for antigen detection, PCR is very reliable. It's a real-time PCR. It's fast and very specific. The virus isolation, and of course, if this is the new outbreak, you need to isolate the virus and make further characterization. ELISA, direct immune staining, agar-gel, immunodiffusion, and patient tests. All these are available. For antibody detection, you can use virus neutralization. Of course, with Rinderpest, you can't do virus neutralization anymore. We cannot work with live virus. There is ELISA's direct immune staining. For histopathology, you could also look and find the intracytoplasmic and intranuclear inclusion bodies and syncytial cells. They're helpful. So there is a Pennside test, and there is a test for each disease. This is using monoclonal antibodies, and this is just the same concept of a Pennside test using monoclonal antibodies, and this is fast test. You can put the sample into the swab, and it's kind of choose migrate. If you see 2 lines, you get positive, and 1 line is negative. these are, as I mentioned, using monoclonal antibodies. Two separate kits, one for Rinderpest and one for PPR. This test can be really a good tool in if you are working in a remote area that you cannot really transmit the samples to the lab. It has about 84% sensitivity and very high specificity, over 90%. It's really never been used for field work for Rinderpest. It's kind of been used a little bit towards the end of the eradication program of Rinderpest. This is not really mass produced to be used right now. So these are selected references. The FAO and OIE are really good sources for information, updated information. And I'd like to thank Dr. White, Kathy, Ethan, and for supplying some of these images. Be glad to answer questions. This is my email address. Questions right
Thanks,
now.
Fawzi. Yeah, we do have a couple questions in the chat. The swine industry has found a new morbillivirus, I'm not saying that correctly, in swine. And they're wondering, what implications do you think this has on likely spread and impact to industry?
Well, I'm not familiar with this report. I know there is recently a report that swine can be affected with PPR. So I'm not sure if it's talking about a different morbillivirus or PPR. But PPR, for many, many years, people think the swine will become a dead-end horse for PPR, but there is a recent paper saying that actually swine can transmit PPR to other animals.
The other question is there any information on how it made such a big jump from Africa to India?
That's really a good question. As we know, these animals, they move really quick from country to country, especially from Africa to Middle East. I'm not sure how it jumped to India, but as I mentioned earlier, that we really don't know if people are becoming aware and discovering the disease in different areas, or is this an actual spread, like moving animals from place to place. So that's a good question. I really don't have a good answer to that.
The next question is, what is the biggest risk of introduction into the U.S., and is the vaccine a DIVA vaccine?
There is a DIVA vaccine, but it's not really available in a mass scale. The vaccines that are now used in Africa is derived from the Nigerian strain 75-1, and it's very effective. It's not a DIVA vaccine. There is also another vaccine used in Asia from lineage 4. It's also not a DIVA vaccine, but it's mass produced and it is very effective vaccine. DIVA vaccine would be nice, but it's not really required. It will help in the outbreak. We didn't really have a DIVA vaccine for Rinderpest either, but the disease has been eradicated. What's the other part of the question, sorry?
What's the biggest risk of introduction to the U.S.?
That's a good question. Well, most outbreaks actually happen when a live animal being introduced, a live infected animal being introduced. I don't see that happening in the U.S., but you really can never say never. As we know, the virus doesn't really survive long outside of the host, which is a big advantage. So unless someone deliberately bring it into this country, I don't really know. The possibility of that, but that's really a good question.
All right, do we have any additional questions? I don't see anything in the chat. I would like to thank everybody for joining today. And just to let everybody know, we do have 2 additional webinars next week. We're going to start off with Dr. Jim Ross, who's presenting on developing, refining, and exercise the Iowa Emergency FMD Vaccination Plan on July 13th at 11 a.m. Eastern Standard Time. And our second webinar next week will be presented by Stephen Wright, the Director of Food Processing for Ceralex, and Jeff Odell, the Technical Director of Marketing Manager for Atlantic Disinfectants Business Line. They're the speakers, and the topic is decontamination in harvest, the 7 steps of sanitation. So hopefully you will be able to join us for those 2 webinars. And with that, I bid you all a great afternoon, and hopefully we'll see you all next week.